Okay, so in this chapter 24, we're going to talk about structure and function of the kidney. So just general functions of the kidney, we know that it acts to filter blood, and by filtering blood, it can help remove water-soluble wastes, as well as control blood pressure by regulating blood volume, right? Like you can filter more blood to get rid of excess water, which would decrease your blood volume and therefore decrease blood pressure. But we also know that the kidneys release different hormones, right? Like the kidneys can release erythropoietin to regulate red blood cell levels. We know the kidneys release renin to regulate blood pressure and also helps to convert vitamin D to regulate blood calcium levels. So this process of filtration just occurs at the renal corpuscle. And the renal corpuscle is going to be essentially a place where the nephron surrounds the glomerular capillaries. And these glomerular capillary cells sit along a basement membrane that acts as a filter. And then you have what's called Bowman's capsule, which is part of your glomerular capsule that sits along the same basement membrane. So what happens then is that these glomerular capillaries protrude into a double layer membrane, kind of like serous membrane, except for when filtration occurs, essentially that fluid gets trapped into a tubule system rather than entering the tissue fluid and then getting picked up by lymph. So fluid gets filtered across this basement membrane between these things called pseudopods, which are essentially just an outgrowth of glomerular capillaries. So if you look up here, what we find then is our glomerular capillaries. This is going to be microscopically small. Now what happens is an afferent arteriole, which is going to carry blood from the cortical radiate artery of your kidney, delivers blood to the glomerular capillaries. Now, as blood circulates through the capillary, some of that fluid is going to get forced out of the capillary wall through bulk flow or hydrostatic pressure, and it gets filtered and enters the space here, which is part of the nephron, and then the remaining blood leaves the glomerular capillary through what's called the efferent arteriole, which then are going to enter the rest of your vein system of the kidney. Now, if we look here at the level of filtration, we know that it occurs at the level of capillaries within the renal corpuscle. So all of this is the renal corpuscle, and it's surrounded by Bowman's capsule or the glomerular capsule. So what's interesting then here is that as fluid leaks out of these capillaries, it's actually picked up by this tubule system rather than just entering the tissue of your kidney itself. And therefore the fluid here that gets filtered, we call it filtrate, can move along the tubule system, and it's modified all along the length of those tubules, which is what we call the nephron. And if you really zoom in on this filtration membrane, you find that it's made of three major things. We have a fenestrated endothelium of capillaries, and this is going to have little holes or fenestrations which allow small substances to pass through. We have a basement membrane which these endothelial cells attach to, and that also serves as a filter because it's a lot of connective tissue fibers that help to trap larger debris, but then allow smaller substances like water, ions, and small wastes to cross. And the outside here is also surrounded by an extra layer of what we call podocytes. Podocyte literally means foot cell, and these podocytes essentially surround the outside of capillaries, and they can modify the leakiness of this membrane because there's spaces between these podocytes called filtration slits, and if these podocytic foot processes separate, the filtration slits get wider and filtrate can enter in more easily. So podocytes can modify the rate of filtration here. Now if you look at a cross section here of the membrane, remember it's made of a fenestrated endothelium, basement membrane, as well as the podocytes. And you can see that smaller substances are able to cross this filtration membrane and then essentially enter the nephron, but then larger things are left in the blood, right? Like entire blood cells, platelets, large proteins, those stay in blood because they're too large to get filtered. Only ultrafiltrate, which is going to be basically very, very small molecules, are able to get forced across this filtration membrane. Now, what determines the direction of this arrow here, filtration, is all about pressure. So what we know then is that there's actually some blood pressure here, or capillary hydrostatic pressure, which we learned about in previous chapters. And that fluid pressure is going to force fluid from higher to lower pressure within the nephron. And that becomes a filtrate. Now it has kind of this yellow tinge to it because there's no blood cells, right? It's just going to be mostly water with a lot of wastes and some ions. Now, it has kind of a yellowish tinge because of the nitrogenous wastes, and this gets concentrated into urine throughout the length of that nephron. Now, there's really three components of urine formation. We have filtration, which only occurs at the glomerulus, the glomerular capillaries, are where filtration occurs. And then as that filtrate moves through the tubule system of your nephron, some of the substances can either be reabsorbed or they can be secreted. Now, if substances are reabsorbed, we're saying that you're moving substances, like ions or even water, from the tubular fluid or filtrate back into your bloodstream. And this can be passive or active transport, but reabsorption is actually reabsorbing it back into blood. That way it's not lost into urine. Because you can imagine that there's certain things that are filtered, like sugar, different ions, even water, that you might not want to lose into urine. Now, secretion is the opposite of reabsorption. Secretion is a process where you're actually taking substances from blood and then transporting them into the filtrate. That way they do get incorporated into urine. So things get filtered, and then for those things that are filtered, some can be reabsorbed, and then other things that aren't filtered, you can also secrete into the tubular fluid. So glomerular filtration rate just refers to the amount of filtrate we can produce in a given minute. And it turns out that our normal glomerular filtration rate is about 125 milliliters per minute. So this indicates normal values, but if it's too low, that can indicate disease. So this discussion question here says, how would it change if your efferent arterioles constricted? Remember, the efferent arterioles are what carry blood away from the glomerulus. So if these efferent arterioles constrict, blood's going to back up into the glomerular capillaries and increase capillary hydrostatic pressure. And if you increase capillary hydrostatic pressure, you're going to actually filter more. So constriction of the efferent arterioles would actually increase glomerular filtration rate. And if you constrict the afferent arterioles, you're going to deliver less blood to the glomerular capillaries. Because remember, afferent arterioles bring blood to the glomerulus. So if these constrict, you're going to have a decrease in blood flow downstream, right, which is the glomerular capillaries. So it's going to decrease glomerular filtration rate. Now, if your blood pressure is low, that's also going to decrease glomerular filtration rate because you need pressure to force that fluid across the filtration membrane. So this next question also says, which of the following would increase glomerular filtration rate, or GFR? Epinephrine, prostaglandins, nitric oxide, or endothelin? Well, we know that substances that would increase your blood pressure should increase GFR, right? So we know that epinephrine is a pretty potent vasoconstrictor. And if you vasoconstrict in the periphery, that's going to increase blood pressure, which should increase GFR. Prostaglandins, we talked about, were vasodilators. Same with nitric oxide. Now, if you vasodilate in the peripheral arteries, that would decrease blood pressure and decrease glomerular filtration rate. And then endothelin is also a vasoconstrictor, so this could actually increase GFR. Now, the problem though is we really want to know, what's happening at the level of efferent and afferent arterioles to precisely kind of get a better understanding of what GFR is going to be. So if you vasoconstrict the afferent arteriole, even if your blood pressure is high, this is going to lead to a decrease in blood flow downstream so that you actually get a lower GFR. And if you vasoconstrict the efferent arteriole, then that's going to cause blood to back up in the glomerular capillaries. And even if your blood pressure is low, that can still potentially increase GFR. So this question here says true or false. If GFR increases, urine output will decrease. Well, that's definitely false, right? Because if you can filter more, then you're going to be making more urine. So higher GFR indicates higher urine output. And this is an interesting phenomenon here, right? And actually there's indirect and direct mechanisms of urine formation. But the direct mechanism here is we're saying that if your blood pressure rises, you can increase GFR, which causes you to eliminate more water through your urine, which is going to increase urine outflow or output. And that's going to decrease your blood volume, which then decreases blood pressure. So there's an autoregulatory mechanism of your kidneys where if your blood pressure is too high, they're just going to filter more and make more urine, which is going to automatically decrease your blood pressure by decreasing blood volume. And the opposite's true if you have low blood pressure. If your blood pressure is too low, then you're not going to filter as much, which means you're going to have a decrease in urine output. So that's going to stabilize blood volume and maybe even increase blood volume, which would increase blood pressure. So the three components of urine formation we talked about were filtration, reabsorption, and secretion. And what we're seeing here is basically the nephron, part of it rather. We have the glomerulus and the proximal convoluted tubule, and that's associated with some nearby blood vessels. Now, we know that filtration occurs at the level of the glomerular capillaries, and that's dependent based on the structure of the filtration membrane and how much pressure is in these vessels. And once those substances get filtered, and they're filtered indiscriminately other than size. So even nutrients that you might not want to lose into urine get filtered if they're small enough, things like glucose, which means that you want to reabsorb some of these substances in the blood to prevent them from getting lost into urine. Now, other things you might want to actually secrete out of blood into the filtrate to lose them into urine. So these are really the three major processes of urine formation. Now, both reabsorption and secretion are under the control of hormones. So there's hormones that influence reabsorption of certain molecules, as is there are hormones that influence secretion of other molecules. So when we talk about reabsorbing water, this is an extremely important aspect of urine formation because your kidneys filter your entire blood volume like once every several minutes. And if you didn't reabsorb water, then essentially you would lose your blood volume extremely quickly. So urine is ultimately like less than 1% of what's originally filtered. And to have such a small volume of urine, we have to reabsorb a lot of water. So most water reabsorption happens in the inner layer of the kidney. We call this the medulla, right? It's deeper. And the medulla is significant because it has a higher osmolarity, so there's more salt in the tissue of the medulla. And that salt is going to pull water out of the nephron back into the tissue fluid, which then can get reabsorbed back into blood. Now, the way the medulla becomes salty or has a high osmolarity is that the nephron loop, or loop of Henle, contains ion pumps, and they use ATP to pump sodium out of filtrate back into the medulla, making it more salty, and then water follows via osmosis. So by reabsorbing ions like sodium, potassium, chloride, you also reabsorb more water. And so this is an important consideration when you start thinking about diuretics. So you can give someone a diuretic that would actually prevent these ion pumps from functioning. And if you don't reabsorb those ions, then you don't reabsorb water, which means you lose more water, and that could increase urine output, which would decrease blood pressure. Now, in terms of what's going on at the nephron itself, we know that the nephron is made of several key parts. We talked about the first part is the glomerulus, and that's going to be the place where filtration occurs. And then as that filtrate flows to the tubules, it goes to the proximal convoluted tubule. And in the proximal convoluted tubule, like 60% of the sodium automatically gets reabsorbed. You get reabsorption of most glucose and amino acids. And there's a little bit of secretion of acids and bases that can occur here. So like if you're too acidic, then you can get rid of acid. Or if you're too basic, you can get rid of more base through secretion. Now, as that fluid starts to enter the nephron loop, we have the descending limb of the nephron loop as well as the ascending limb. Now, the descending limb is permeable to water. Remember, water gets reabsorbed via osmosis because the tissue out here is more salty, so water follows those solutes and then gets reabsorbed back into the tissue fluid of your kidney. Now, in the thick ascending limb of the loop of Henle, this has a lot of sodium, potassium, and chloride pumps. And so what happens here is we can actually have ATPases, and they use the chemical energy of ATP to pump sodium, potassium, and chloride into the medulla. That makes the medulla more salty, which allows for water to get reabsorbed. So it's important to note then that the ascending limb here sets the stage for water reabsorption. So if you really think about what's happening as the filtrate descends, well, it's getting more concentrated because you're reabsorbing water. So that means that when you get to the tip of the nephron loop, this is going to be the most concentrated. In fact, at this point, it's about 4 times more concentrated than normal blood. And then as that filtrate starts to ascend up the nephron loop, then you start to reabsorb the salt, right? The sodium, potassium, chloride, which means by the time that filtrate gets to the proximal convoluted tubule, what's happening here is actually going to be more dilute. So you have a smaller volume of dilute filtrate. And at the level of the distal convoluted tubule here, this one's under the control of hormones, things like aldosterone, parathyroid hormone, even antidiuretic hormone. And they can influence secretion and reabsorption of different ions. And then once we get to the collecting duct, this is not technically part of the nephron. In fact, lots of nephrons will connect up with the same collecting duct. But the collecting duct is significant because this is under the influence of antidiuretic hormone, so that the effect of ADH on this part of your nephron causes you to reabsorb more water. And so that as long as ADH is present, you're going to make a smaller volume of highly concentrated urine by the time that fluid leaves the nephron. So in terms of reabsorption of water, we talked about how sodium and chloride are pumped into the medulla, which make the fluid of the medulla more salty or have a higher osmolarity. And we say that it's hypertonic, right? Now, because it's hypertonic, water is going to follow that hypertonic solution that you find inside of the kidney tissue fluid of the medulla. And so water gets reabsorbed from the nephron itself, or even from the collecting duct, like from that filtrate back into the tissue fluid. And then from there, it can enter in your bloodstream again. And this is a very important aspect because we wouldn't want to lose all of our water into our urine because you would dehydrate extremely fast. So then that drugs that make your medulla less salty prevent you from reabsorbing water, which then would cause you to lose more water into urine. So this question here says, which renal structure reabsorbs water? Is it the proximal loop, distal loop, the collecting duct, or the glomerulus? Well, most water, at least of those that are listed, get reabsorbed by the collecting duct. Now, a lot of water gets reabsorbed by the descending limb of the nephron loop, but that wasn't listed. Now, the collecting duct is the next best answer of the ones that are listed, and this is going to be associated with the reabsorption of water, but this is under the control of antidiuretic hormone. And so in the absence of antidiuretic hormone, then you won't reabsorb as much water, so you're going to make a bigger volume of dilute urine. But if antidiuretic hormone is present, and it's going to stimulate the cells of your collecting duct to reabsorb more water, then you're going to make a smaller volume of highly concentrated urine. Now, the amount of water that's reabsorbed depends on how much can move out of the collecting duct and back into your kidney tissue fluid of the medulla. And we talked about how this is dependent on antidiuretic hormone, or ADH. We learned earlier that antidiuretic hormone comes from the posterior portion of the pituitary gland, and it's a neuropeptide that's secreted into the bloodstream. Now, antidiuretic hormone causes you to reabsorb more water, so it would make sense then that you would release ADH under conditions where either you have low blood pressure or hyperosmolarity of the blood, which means it's too salty, and so you can reabsorb more water to get your blood volume up to increase blood pressure, or reabsorb more water to make your blood less salty. So you might wonder, well, what stimulates the release of ADH? Well, we said that ADH was part of the RAAS, or the Renin-Angiotensin-Aldosterone system. So antidiuretic hormone, or vasopressin, was one of the basically one of the hormones that's released in response to angiotensin II. Now, antidiuretic hormones also stimulate to be released when hypothalamic osmoreceptors sense that your blood is too salty. Now, ADH makes the collecting duct more permeable to water, so more water can be reabsorbed from the urine back into blood. Now, it's not correct to say from urine into blood. It's more correct to say from filtrate into blood, because filtrate is what you find in the nephron and the collecting duct. Once that fluid enters the calyces of your kidney, then it's technically urine and it can't be modified any further. But it's the same fluid that will become urine later. Now, the blood osmolarity decreases when you reabsorb water because your blood becomes less salty when it's more diluted. So this question here says, true or false? An increase in ADH decreases urine output. And that's absolutely true. We said that ADH is antidiuretic hormone, makes the collecting duct more permeable to water, so that water can leave the duct and get reabsorbed back into blood. And if you reabsorb that water, it won't end up into urine, so you're going to have less urine being produced. Now, when urine reaches the distal convoluted tubule, which is the last part of your nephron, we say that cells of the distal convoluted tubule, we call these the juxtaglomerular cells, they can actually measure flow in the afferent arteriole, and they also get information from cells called macula densa that measure how salty the fluid is. And if the fluid is too salty, or if your blood pressure is too low, these cells can release a hormone called renin, and they're released by cells called granular cells of this juxtaglomerular complex. But what renin does is it helps to, basically goes in the bloodstream. It helps to convert angiotensinogen into angiotensin 1, and then angiotensin 1 is converted to 2 by ACE in the lungs. And then in turn, that causes you to reabsorb more sodium and more water because you're going to release aldosterone as well as antidiuretic hormone. And then we also get the release of potassium or secretion of potassium because one of the effects of aldosterone is not just to reabsorb sodium, but you also secrete potassium. So if you had chronic hypotension, that could potentially cause hypokalemia through excessive potassium secretion. Now, we said that the Renin-Angiotensin-Aldosterone pathway is essentially where, when your blood pressure is low, you get the release of renin that ultimately leads to the production of angiotensin II. Now, when angiotensin II stimulates the release of aldosterone and ADH, it causes the reabsorption of sodium and water, which raises your blood volume. It won't have a major effect on osmolarity because you're reabsorbing both sodium and water, but it's going to increase blood pressure. But we learned that aldosterone also causes potassium secretion simultaneously. In fact, this is an exchanger. You can't reabsorb sodium via aldosterone without also secreting potassium. So if you reabsorb one sodium, you're going to secrete one potassium. And ultimately, over time, that could potentially lead to very low levels of potassium in the blood. So this discussion question here says, which changes would increase the release of renin? Well, we learned that renin release was stimulated by low blood pressure. So if you have an increase in blood volume, then your blood pressure would be high, so you wouldn't want to release renin because that would make your blood pressure even higher. So definitely a decrease in blood volume would stimulate the release of renin. And then if your blood pressure is already high, again, you wouldn't want to release renin, but a decrease in blood pressure, you would want to release renin to get your blood pressure back up. Because remember, renin leads to the production of angiotensin II, and angiotensin II is both a vasoconstrictor, which would increase blood pressure, and it stimulates the release of hormones that ultimately cause you to have a higher blood pressure. So of these four, really what would cause renin secretion is either a decrease in blood volume or a decrease in blood pressure. Both of these would cause renin to be released by your kidneys. Now the scenario here says a man was given a drug that stopped the ion pumps in the loop of Henle. Remember, these ion pumps are what allow you to reabsorb sodium and potassium and chloride. So the question here says, what happened to this individual's medullary osmolarity? Well, if they're not reabsorbing these salts, then the medulla will become less salty or have a decrease in osmolarity. And they're going to find that there's going to be more sodium left over in the filtrate. So there's more sodium in the distal convoluted tubule. You can't reabsorb water without reabsorbing the sodium because water is only reabsorbed via osmosis. And if you have a lot of sodium in the distal convoluted tubule, that's going to cause the release of renin. And then if you release a lot of renin, that's going to cause you to secrete too much potassium because aldosterone is going to cause you to reabsorb more sodium but also secrete potassium, leading to hypokalemia. So what's weird then is that if you gave someone a drug that stopped the ion pumps in a loop of Henle, they could end up with high urine output and also low blood potassium because of loss of too much of this ion. Now, removing sodium and chloride from water from your blood is an important aspect of blood pressure regulation as well. But the problem, though, is that we have no deliberate way to secrete excess sodium. This is why we want to be careful about eating too much sodium in our diets, because our bodies have no deliberate way to secrete too much sodium. Like if you have too much sodium in your body, there's no way to get rid of that sodium other than to not reabsorb it. So here's the weird part, is if your sodium levels are high, like hypernatremia, you don't have a mechanism in your body, humans don't rather, to deliberately secrete excess sodium. So the only thing we can do is prevent our kidneys from reabsorbing sodium, which means that slowly over time, you're going to lose sodium, but you can't get rid of excess sodium quickly. And this is actually under the control of a hormone called atrial natriuretic peptide. So atrial natriuretic peptide, it's actually released by the atria of your heart, which is kind of interesting. And it's made in response to overstretched atria. And this would actually correlate with high blood pressure or high blood volume. Now, ultimately, atrial natriuretic peptide, once it's released by your heart, it causes the kidneys to stop reabsorbing sodium and chloride, and so that more sodium and chloride are lost into the urine. And if you don't reabsorb these salts, then you can't reabsorb as much water, so it's going to increase urine output, which is going to increase, it's going to decrease blood pressure, and it's going to decrease the stretch on your heart as well as how much work your heart has to produce. So what's really neat is that this is a hormone your heart secretes in response to high blood pressure, which ultimately helps protect the heart itself. So this discussion here says, what will happen to urine if aldosterone is given? Well, remember, aldosterone causes you to reabsorb sodium but secrete potassium. So that means that your urine levels will have low sodium but higher potassium. Or what will happen to urine if your ADH levels are high? Well, we learned that ADH causes you to reabsorb water, so that urine after ADH is being released is going to be more concentrated and a smaller volume because you're reabsorbing more of that water. And this next discussion question says a man has severe renal disease. And this says, why does he develop anemia and weak bones? Well, remember, the kidneys also release erythropoietin. So one of the consequences of kidney disease is renal anemia, which is essentially where your kidneys don't release erythropoietin in response to low blood oxygen. So that ultimately that causes anemia because your kidneys are failing, which is kind of odd. And then you can also see weakening of bones because of the fact that your kidneys activate vitamin D. Now, this question here says, what hormone secreted by the kidney stimulates red blood cell formation in bone marrow? And that was erythropoietin. So erythropoietin literally means producing erythrocytes, and this is actually caused by decreased levels of this hormone that can lead to anemia. But if you have too much erythropoietin, it can lead to what's called polycythemia. So we know one of the normal functions of the kidneys is to basically eliminate wastes through filtration. And we can also eliminate waste through secretion. So those together would aid in what we call renal clearance. So renal clearance refers to how much blood the kidneys can clean in a minute. There's different ways to measure renal clearance, but ultimately it boils down to how much of a substance is cleared by the kidneys in any given moment of time. So if blood contains 1 milligram of wastes per 100 ml of blood, and the person produces 1 ml of urine per minute, and if that urine contains 1 milligram of waste per ml, you might wonder, well, then what's their glomerular filtration rate, right? Well, let's think about this. We know that the kidneys reabsorb about 99% of the water that's filtered. Now, if you're finding a whole milligram of that waste in the urine for every 1 ml of that urine, but you find 1 milligram of that waste in 100 ml of blood, then you can make a pretty good guess that your kidneys cleared 100 ml of blood in that minute, and that's how it got rid of 100 milligrams of that waste. So this question here says, how much blood did the kidney clean in 1 minute? Well, it should be around 100 ml or so, because you're finding approximately the same amount of that waste in different concentrations, but ultimately it's the same total amount. And what we find then is that the kidneys would have cleared about 100 ml of that blood. Now that would be kind of slow for glomerular filtration rate, because normal glomerular filtration rate is 125 ml per minute, but it's close. So in terms of the kidney functions, we know that the kidneys help to eliminate wastes. And when those wastes build up in the blood, they can cause problems throughout the body, and we'll talk about that in future slides, or next chapter rather. But what percent of the kidney function, if your serum creatinine is like 2, 3, or 10 milligrams per deciliter? Well, we know that creatinine is a metabolite. And as a metabolite, it's basically the product of chemical reactions in your body. And it's a waste as well, and it's eliminated by your kidneys. So we can look at creatinine clearance as a measure of kidney function, because your kidneys should be able to get rid of a certain amount of creatinine per minute. Now, if you start looking at creatinine of the serum, what we're saying then is that if serum creatinine levels start to rise, like if they get higher and higher and higher, that's going to suggest that your kidneys aren't clearing that waste as well if the waste is accumulating within blood, because your kidneys should normally prevent that waste from being too high in concentration within blood. So what these tell us then is if your creatinine levels are rising, then your kidney function is falling. And that's generally true. Now, there are other things that can increase your creatinine levels of your blood, like if you have a protein-rich diet, or if you're taking like creatine supplements, that's going to abnormally increase this metabolite in your blood. But for the most part, under normal circumstances and normal diet, If creatinine levels rise in the blood, that suggests that your kidney function is on the decline. Now, you can find this kind of data out there. Like, we know how much of your kidney is functioning normally if you find this much creatinine in your blood or this much creatinine. And if you find 10 milligrams per deciliter of creatinine in your blood, you're saying that the kidneys are functioning pretty poorly. This is a tremendous amount of this waste. So the kidney function would be like on the order of, probably less than 10% of normal clearance. So in terms of understanding kidney function, all we have to look at then is a proportion or a ratio of normal serum creatinine to current serum creatinine. And if this ratio gets smaller, then that suggests that your kidney function is also on the decline because these wastes are going to build up in your bloodstream. So with normal serum creatinine, it should be fairly low. Like we're saying that it's usually less than a milligram or so. And if your serum creatinine is much higher, then this fraction gets smaller, which means your kidney function is also getting smaller or less. It's less efficient. So this discussion question here says, which of these tests indicate, what do the results indicate? Like if you have proteinuria. Now, severe proteinuria means we have a lot of protein in the urine. Now, we normally don't find protein in urine, at least not in like abundant amounts, because the glomerular filtration membrane isn't permeable to blood protein. Remember, blood proteins are too large and they can't cross that filtration membrane. So proteinuria would actually indicate that your glomerular filtration membrane is damaged, as in the case with like some of the nephropathies that we'll talk about in future chapters. Now, if you're finding red blood cell casts in urine as well, these casts are essentially aggregations of red blood cells and hemoglobin. And we also normally don't find red blood cells in urine, not a lot, because again, red blood cells aren't filtered. So if we're finding a lot of red blood cells in the urine, that's going to indicate either kidney damage, urinary bladder damage, or other damage to other portions of the urinary tract, like the urethra or ureters, because blood can get into the urine in a variety of ways. So it doesn't necessarily suggest kidney damage, but it could indicate that. Now we know that your urine specific gravity is usually around 1, and if it's lower than 1, and if it's much lower than 1, that's going to suggest that your urine is very dilute and there's not a lot of ions. And this could be a problem that's severalfold. It could be that your nephrons aren't adequately absorbing ions, and that's why you're losing a lot of water. Or it could mean that you have fluid overload and your kidneys are just trying to get rid of that excess water, which is going to make your specific gravity of urine lower. And then creatinine and BUN are also indicators of kidney function. Because creatinine, we said, is a metabolite of creatine phosphate, and it's cleared by your kidneys. Now, normal creatinine levels are about 0.6 to 1.2 milligrams per deciliter. So if someone has 6 milligrams per deciliter of creatinine in their blood, that's going to indicate that their kidneys aren't adequately getting rid of that metabolite. So that's going to suggest that their kidneys aren't functioning well, and this might indicate kidney disease. Now, BUN is blood urea nitrogen, and normal BUN levels are around 20 milligrams per deciliter. So this is obviously elevated quite a bit. And like creatinine, this is going to tell us that if blood urea nitrogen is high, that your kidneys are not getting rid of this nitrogenous waste, so it's staying in your blood, which is going to tell us why this is in high concentration within blood. So this is also going to indicate kidney disease. So this last question here says, if 125 mils of ultrafiltrate is formed each minute, how much is formed in a day? Well, we know that actual urine excretion is about 1.5 liters a day, and you reabsorb approximately 99% of the water that's filtered in your kidneys. So if it's 125 mils per minute, you multiply that out times 60 minutes per hour times 24 hours, it ends up being several thousand liters that you're going to filter every day. But the actual amount of urine you're going to produce is only like 1.5 liters. So it's really not that much. The reason for this is that your proximal convoluted tubule, which is the first part of your nephron after the glomerulus, that's going to reabsorb about 60% of all the nutrients and ions that are in that filtrate. Almost immediately, 60% gets reabsorbed. Now this absorbs at an automatic rate, And the amount of solute it can reabsorb is we consider a transport maximum. Now it's possible to reach transport maximum, and we'll talk about this when we get to diabetes, when we talk about things like glycosuria. But if nutrients don't get reabsorbed because you've reached your transport maximum, then they might end up in your urine, and that's not good because then you're talking about losing nutrients in the urine. But typically you reabsorb all those nutrients as long as your kidneys are functioning well. Now, I'm not saying here that we only absorb 60% of filtered. It's just that the proximal convoluted tubule reabsorbs 60% of filtrate. Otherwise, the nephron loop reabsorbs quite a bit. The distal convoluted tubule can reabsorb quite a bit. Even the collecting duct can reabsorb quite a bit. And so the actual filtrate or tubular fluid that later becomes urine is like less than 1% of what was originally filtered. And this is because we're going to want to bring this back into our bloodstream so we don't just lose it from our body, because we obviously don't have thousands of liters to lose every day.